Section 8.3, Extensions of the Laws of Inheritance. By the end of this section, you will be able to identify non-Mendelian inheritance patterns such as incomplete dominance, codominance, multiple alleles, and sex linkage from the results of crosses. Explain the effect of linkage and recombination on gamete genotypes, and explain the phenotypic outcomes of epistatic effects among genes. Mendel studied traits with only one mode of inheritance in pea plants. The inheritance of the traits he studied all followed the relatively simple pattern of dominant and recessive alleles for a single characteristic. There are several important modes of inheritance discovered after Mendel's work that do not follow the dominant and recessive single gene model. Alternatives to dominance and recessiveness. Mendel's experiments with pea plants suggested that 1. Two types of units, or alleles, exist for every gene. 2. Alleles maintain their integrity in each generation, no blending. And 3. In the presence of the dominant allele, the recessive allele is hidden with no contribution to the phenotype. Therefore, recessive alleles can be carried and not expressed by individuals. Such heterozygous individuals are sometimes referred to as carriers. Since then, genetic studies in other organisms have shown that much more complexity exists, but that the fundamental principles of Mendelian genetics still hold true. In the sections to follow, we consider some of the extensions of Mendelism. Incomplete dominance. Mendel's results, demonstrating that traits are inherited as dominant and recessive pairs, contradicted the view at the time that offspring exhibited a blend of their parents' traits. However, the heterozygote phenotype occasionally does appear to be intermediate between the two parents. For example, in the snapdragon, Antirrhinum majus, a cross between a homozygous parent with white flowers and a homozygous parent with red flowers will produce offspring with pink flowers. Note that different genotypic abbreviations are used for Mendelian extensions to distinguish these patterns from simple dominance and recessiveness. This pattern of inheritance is described as incomplete dominance, meaning that one of the alleles appears in the phenotype in the heterozygote, but not to the exclusion of the other, which can also be seen. The allele for red flowers is incompletely dominant over the allele for white flowers. However, the results of a heterozygote self-cross can still be predicted, just as with Mendelian dominant and recessive crosses. In this case, the genotypic ratio would be 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive, and the phenotypic ratio would be 1 to 2 to 1 for red to pink to white. The basis for the intermediate color in the heterozygote is simply that the pigment produced by the red allele, anthocyanin, is diluted in the heterozygote and therefore appears pink because of the white background of the flower petals. Figure 8.12. These pink flowers of a heterozygote snapdragon result from incomplete dominance. Codominance. A variation on incomplete dominance is codominance, in which both alleles for the same characteristic are simultaneously expressed in the heterozygote. An example of codominance occurs in the ABO blood groups of humans. The A and B alleles are expressed in the form of A or B molecules present on the surface of red blood cells. Homozygotes, for A and B, express either the A or the B phenotype, and heterozygotes express both phenotypes equally. The heterozygote individual has blood type AB. In a self-cross between heterozygotes expressing a codominant trait, the 3 possible offspring genotypes are phenotypically distinct. However, the 1 to 2 to 1 genotypic ratio of a Mendelian monohybrid cross still applies. Figure 8.13. This Punnett square shows an AB-AB blood type cross. Multiple alleles. Mendel implied that only two alleles, one dominant and one recessive, could exist for a given gene. We now know that this is an oversimplification. Although individual humans and all diploid organisms can only have two alleles for a given gene, multiple alleles may exist at the population level, such that many combinations of two alleles are observed. Note that when many alleles exist for the same gene, the convention is to denote the most common phenotype or genotype in the natural population as the wild type, often abbreviated with a plus symbol. All other phenotypes or genotypes are considered variants or mutants of this typical form, meaning they deviate from the wild type. The variant may be recessive or dominant to the wild type allele. An example of multiple alleles is the ABO blood type system in humans. In this case, there are three alleles circulating in the population. The dominant A allele codes for A molecules in the red blood cells, or on the red blood cells. The dominant B allele codes for B molecules on the surface of red blood cells, and the recessive I allele codes for no molecules on the red blood cells. In this case, the dominant A and B alleles are codominant with each other and are both dominant over the I allele. Although there are three alleles present in a population, each individual only gets two of the alleles from their parents. This produces the genotypes and phenotypes shown in figure 8.14. Notice that instead of three genotypes, there are six different genotypes when there are three alleles. The number of possible phenotypes depends on the dominance relationship between the three alleles. Figure 8.14. Inheritance of the ABO blood system in humans is shown. Evolution connection. Multiple alleles confer drug resistance in the malaria parasite. Malaria is a parasitic disease in humans that is transmitted by infected female mosquitoes, including Anopheles gambiae, and is characterized by cyclic, high fevers, chills, flu-like symptoms, and severe anemia. Plasmodium falciparum and P. vivax are the most common causative agents of malaria, and P. falciparum is the most deadly. When promptly and correctly treated, P. falciparum malaria has a mortality rate of 0.1%. However, in some parts of the world, the parasite has evolved resistance to commonly used malaria treatments, so the most effective malarial treatments can vary by geographic region. 90% of malaria victims live in Africa, most of them children under the age of 5. In Southeast Asia, Africa, and South America, P. falciparum has developed resistance to the anti-malarial drugs chloroquine, mefloquine, and sulfadoxine pyrimethamine. P. falciparum, which is haploid during the life stage in which it is infective to humans, has evolved multiple drug-resistant mutant alleles of the DHPS gene. Varying degrees of sulfadoxine resistance are associated with each of these alleles. Being haploid, P. falciparum needs only one drug-resistant allele to express this trait. In Southeast Asia, different sulfadoxine-resistant alleles of the DHPS gene are localized to different geographic regions. This is a common evolutionary phenomenon that comes about because drug-resistant mutants arise in a population and interbreed with other P. falciparum isolates in close proximity. Sulfadoxine-resistant parasites cause considerable human hardship in regions in which this drug is widely used as an over-the-counter malaria remedy. As is common with pathogens that multiply to large numbers with an infection cycle, P. falciparum evolves relatively rapidly, over a decade or so, in response to the selective pressure of commonly used anti-malarial drugs. For this reason, scientists must constantly work to develop new drugs or drug combinations to combat the worldwide malaria burden. In late 2021, R21 Matrix M became the first vaccine to be recommended for widespread use by the World Health Organization. At least 10 other candidate vaccines are in development. The effort is an multinational one involving governments, universities, nonprofits, philanthropists, and pharmaceutical companies. Much of the recent progress can be credited to organizations within the most affected countries, such as the Malaria Research and Training Center in Mali. Founded by Ogobara Duombo and Yaya Toure in the 1990s, the center has emerged as a primary frontline research driver, including running many of the critical clinical trials that are so important to vaccine development and approval. Sex-linked traits. In humans, as well as in many other animals and some plants, the sex of the individual is determined by sex chromosomes, one pair of non-homologous chromosomes. Humans may identify as being male, female, neither of these, both, or other genders independently of these chromosomes, but the sex chromosomes can be associated with certain traits. Until now, we have only considered inheritance patterns among non-sex chromosomes or autosomes. In addition to 22 homologous pairs of autosomes, human females have a homologous pair of X chromosomes, whereas human males have an XY chromosome pair. Although the Y chromosome contains a small region of similarity to the X chromosome so that they can pair during meiosis, the Y chromosome is much shorter and contains fewer genes. When a gene being examined is present on the X but not the Y chromosome, it is X-linked. Eye color in Drosophila, the common fruit fly, was the first X-linked trait to be identified. Thomas Hunt Morgan mapped this trait to the X chromosome in 1910. Like humans, Drosophila males have an XY chromosome pair and females are XX. In flies, the wild-type eye color is red and is dominant to white eye color. Because of the location of the eye color gene, reciprocal crosses do not produce the same offspring ratios. Males are said to be hemizygous in that they have only one allele for any X-linked characteristic. Hemizygosity makes descriptions of dominance and recessiveness irrelevant for XY males. Drosophila males lack the white gene on the Y chromosome. That is, their genotype can only be white or red, dominant or recessive. In contrast, females have two allele copies of this gene and can be homozygous dominant, heterozygous, or homozygous recessive. Figure 8.15. In Drosophila, the gene for eye color is located on the X chromosome. Red eye color is wild type and is dominant to white eye color. In an X-linked cross, the genotypes of F1 and F2 offspring depend on whether the recessive trait was expressed by the male or the female in the P region, the P generation. With respect to Drosophila eye color, when the P male expresses the white eye phenotype and the female is homozygously red-eyed, all members of the F1 generation exhibit red eyes. The F1 females are heterozygous, and the males are all hemizygous dominant, having received their X chromosome from the homozygous dominant P female and the Y chromosome from the P male. A subsequent cross between the heterozygous female and the hemizygous dominant male would produce only red-eyed females with homozygous dominant or heterozygous genotypes. In both red and white-eyed males with hemizygous dominant or hemizygous recessive genotypes. Now, consider a cross between a homozygous white-eyed female and a male with red eyes. The F1 generation would exhibit only heterozygous red-eyed females and only white-eyed males. Half of the F2 females would be red-eyed, heterozygous, and half would be white-eyed, homozygous recessive. Similarly, half of the F2 males would be red-eyed, hemizygous dominant, and half would be white-eyed, hemizygous recessive. Figure 8.16. Crosses involving sex-linked traits often give rise to different phenotypes for the different sexes of offspring. as is the case for this cross involving red and white eye color in Drosophila. In the diagram, recessive is the white eye mutant allele, and dominant is the wild type red eye allele. Discoveries in fruit fly genetics can be applied to human genetics. When a female parent is homozygous for a recessive X-linked trait, the parent will pass the trait on to 100% of the male offspring because the males will receive the Y chromosome from the male parent. In humans, the alleles for certain conditions, some color blindness, hemophilia, and muscular dystrophy, are X-linked. Females who are heterozygous for these diseases are said to be carriers and may not exhibit any phenotypic effects. These females will pass the disease to half of their male offspring and will pass carrier status to half of their female offspring. Therefore, X-linked traits appear more frequently in males than females. In some groups of organisms with sex chromosomes, the sex with the non-homologous sex chromosomes is the female rather than the male. This is the case for all birds. In this case, sex-linked traits will be more likely to appear in the female in whom they are hemizygous. Linked genes violate the law of independent assortment. Although all of Mendel's pea plant characteristics behaved according to the law of independent assortment, we now know that some allele combinations are not inherited independently of each other. Genes that are located on separate, non-homologous chromosomes will always sort independently. However, each chromosome contains hundreds or thousands of genes, organized linearly on chromosomes like beads on a string. The segregation of alleles into gametes can be influenced by linkage, in which genes that are located physically close to each other on the same chromosome are more likely to be inherited as a pair. However, because of the process of recombination or crossover, it is possible for two genes on the same chromosome to behave independently, or as if they are not linked. To understand this, let us consider the biological basis of gene linkage and recombination. Homologous chromosomes possess the same genes in the same order, though the specific alleles of the gene can be different on each of the two chromosomes. Recall that during interphase and prophase 1 of meiosis, homologous chromosomes first replicate and then synapse, with like genes on the homologs aligning with each other. At this stage, segments of homologous chromosomes exchange linear segments of genetic material. This process is called recombination or crossover, and it is a common genetic process. Because the genes are aligned during recombination, the gene order is not altered. Instead, the result of recombination is that maternal and paternal alleles are combined onto the same chromosome. Across a given chromosome, several recombination events may occur, causing extensive shuffling of alleles. Figure 8.17. The process of crossover, or recombination, occurs when two homologous chromosomes align and exchange a segment of genetic material. When two genes are located on the same chromosome, they are considered linked, and their alleles tend to be transmitted through meiosis together. To exemplify this, imagine a dihybrid cross involving flower color and plant height, in which the genes are next to each other on the chromosome. If one homologous chromosome has alleles for tall plants and red flowers, and the other chromosome has genes for short plants and yellow flowers, then when the gametes are formed, the tall and red alleles will tend to go together into a gamete, and the short and yellow alleles will go into the other gamete. These are called the parental genotypes because they have been inherited intact from the parents of the individual producing gametes. But unlike if the genes were on different chromosomes, there will be no gametes with tall and yellow alleles and no gametes with short and red alleles. If you create a Punnett square with these gametes, you will see that the classical Mendelian prediction of 9 to 3 to 3 to 1 outcome of a dihybrid cross would not apply. As the distance between two genes increases, the probability of one or more crossovers between them increases, and the genes behave more like they are on separate chromosomes. Geneticists have used the proportion of recombinant gametes, the ones not like the parents, as a measure of how far apart genes are on a chromosome. Using this information, they have constructed linkage maps of genes on chromosomes for well-studied organisms, including humans. Mendel's seminal publication makes no mention of linkage, and many researchers have questioned whether he encountered linkage but chose not to publish those crosses out of concern that they would invalidate his independent assortment postulate. The garden pea has 7 pairs of chromosomes, and some have suggested that his choice of 7 characteristics was not a coincidence. However, even if the genes he examined were not located on separate chromosomes, it is possible that he simply did not observe linkage because of the extensive shuffling effects of recombination. Epistasis. Mendel's studies in pea plants implied that the sum of an individual's phenotype was controlled by genes, or as he called them, unit factors, such that every characteristic was distinctly and completely controlled by a single gene. In fact, single observable characteristics are almost always under the influence of multiple genes, each with 2 or more alleles acting in unison. For example, at least 8 genes contribute to human eye color. In some cases, several genes can contribute to aspects of a common phenotype without their gene products ever directly interacting. In the case of organ development, for instance, genes may be expressed sequentially, with each gene adding to the complexity and specificity of the organ. Genes may function in complementary or synergistic fashions, such that 2 or more genes expressed simultaneously affect a genotype. An apparent example of this occurs with human skin color, which appears to involve the action of at least 3 and probably more genes. Cases in which inheritance for a characteristic like skin color or human height depend on the combined effects of numerous genes are called polygenic inheritance. Genes may also oppose each other, with one gene suppressing the expression of another. In epistasis, the interaction between genes is antagonistic, such that one gene masks or interferes with the expression of another. Epistasis is a word composed of Greek roots meaning standing upon. The alleles that are being masked or silenced are said to be hypostatic to the epistatic alleles that are doing the masking. Often, the biochemical basis of epistasis is a gene pathway in which expression of one gene is dependent on the function of a gene that precedes or follows it in the pathway. An example of epistasis is pigmentation in mice. The wild-type coat color, agouti, is dominant to solid-colored fur. However, a separate gene, C, when present as the recessive homozygote, negates any expression of pigment from the A gene, the dominant A gene, and results in an albino mouse. Therefore, the genotypes homozygous dominant A, homozygous recessive C, heterozygous A, homozygous dominant C, and homozygous AC all produce homozygous recessive AC all produce the same albino phenotype. A cross between heterozygotes for both genes would generate offspring with a phenotypic ratio of 9 agouti to 3 black to 4 albino. In this case, the C gene is epistatic to the A gene. Figure 8.18. In this example of epistasis, one gene, C, masks the expression of another, A, for coat color. When the C allele is present, coat color is expressed. When it is absent, no coat color is expressed. Coat color depends on the A gene, which shows dominance, with the recessive homozygote showing a different phenotype than the heterozygote or dominant homozygote.